And now it is my pleasure to introduce our first set of presenters for today. First, we will be hearing from Dr. Tahsin Mirza, Associate Director for Regulatory Affairs from the Office of Study Integrity and Surveillance within the Office of Translational Sciences. Next, we will hear from Dr. Gabriel Davila, also a biologist also from the Office of Study Integrity and Surveillance. And then that will be followed by Dr. Liang or Paul Ye, an interdisciplinary scientist within the Division of New Drug Study Integrity, also within the Office of Study Integrity and Surveillance. So please join me in welcoming our first presentation and presenter, Dr. Mirza.
Greeting, folks. My name is Tahsin Mirza. I serve as the Associate Director for Regulatory Affairs in the Office of Study Integrity and Surveillance, or OSIS. Please note that this presentation reflects the views of this author. It should not be construed to represent FDA's views or policies. In the next few minutes, I would like to explain the mission, vision of OSIS and give you a broader picture of the three compliance programs or CPs that fall under its purview. This current and the next section will provide in-depth analysis of each of our compliance programs. This presentation will go over the organization structure, roles and responsibilities, and the 4 steps OSIS follows in order to perform our core mission by following 4 steps: select, inspect, report, and support. OSIS selects sites for inspection, inspects study conduct and data reliability, reports on inspections through establishment inspection reports or EIRs, and finally supports stakeholders with recommendations and evaluations. Our stakeholders being the various review divisions within CDER. OSIS resides under US FDA's Center for Drug Evaluation and Research, or CDER. Under CDER, there are several super offices. OSIS is part of the super office of Translation Sciences, Office of Translation Sciences, that is. And OSIS has an immediate office to support and oversee the operation of 2 divisions, which are Division of New Drugs Study Integrity, DNDSI, and Division of Generic Study Integrity, or DGDSI. OSIS's vision is to improve the public health by protecting study subjects and promoting properly conducted studies. And OSIS's mission is to promote the public health by ensuring the welfare of study subjects and by verifying the quality, study integrity, and regulatory compliance of bioavailability, bioequivalence, or BABE, nonclinical GLP, and animal rule AR studies. Please remember the 3 major types of studies that are under the purview of OSIS from this slide, which are BABE. Under BABE, both analytical and clinical portions. Number 2, nonclinical GLP, and number 3, animal rule studies. OSIS supports CDER FDA's mission by serving as one of the last sets of eyes for authenticating data and to provide assurance that the data supporting regulatory decisions are reliable. It does so by following 4 distinct steps as mentioned before: select, inspect, report, and support. The rest of this presentation describes each of these steps. The first core function or step in site evaluations is select. OSIS selects sites that conduct pharmacokinetic PK bioavailability, bioequivalence, BABE, good lab practice, GLP, and animal rule, AR studies, it does so for doing what? To perform study-directed and comprehensive surveillance inspections of sites. The keyword is study-directed. The firm must submit a study in a drug application for OSIS to select a site for an inspection. OSIS uses a risk-based site selection process. Sites are prioritized for inspection based on several criteria that may include time since last inspection, outcome of past inspections and corrective actions taken, number of pending applications, and dates of study conduct in the pending applications compared to those reviewed in previous inspections. Based on risk assessment, the inspector in charge of the inspection may choose to audit parts of several studies as opposed to deeper evaluation of fewer studies. When issues are identified, a determination is made if it is isolated or a systemic issue affecting multiple studies. This allows OSIS to better assess the overall performance of the site. The great majority, more than 80%, of the site assessments submitted to OSIS for inspection are involved in ANDA studies. The next category is NDAs and then BLAs. That brings us to the next step in the process after site selection. It is to inspect. Inspection of a site is conducted in order to ensure the quality and integrity of studies submitted in drug applications. OSIS assigns and conducts inspections in collaboration with the Office of Regulatory Affairs. Additionally, OSIS is responsible for investigating allegations and complaints of regulatory and non-compliance. OSIS relies on compliance programs, or CPs in short, to serve as a guide for conducting activities to evaluate industry compliance with FD&C Act and other applicable laws. Compliance programs provide a set of instructions to FDA personnel for conducting inspections. The CPs under the purview of OSIS are In Vivo BABE studies, the clinical portion, In Vivo BABE studies, the analytical portion, inspection of non-clinical labs conducting animals rule-specific studies, and finally, the good lab practice non-clinical. This workshop will provide an in-depth view of each of the compliance programs with specific cases on day 2. The scope of an OSIS inspection depends on the compliance program involved. Some of the major elements are provided on this slide, which include facilities and site operations, drug product and subject sample accountability, which includes storage, handling, and processing, reserve samples, SOPs, protocols and protocol deviations, training records, method validations and sample analysis, method performance, audit trails and data security, instrument calibration and maintenance, documentation, AE reporting, monitor reports, and IRB/IEC oversight, specifically in case of clinical inspections. Study data integrity is the cornerstone of FDA's ability to protect the public health. It's a part of OSIS's core mission, and it's emphasized in the name itself, Office of Study Integrity and Surveillance, Study Integrity. Data integrity provides assurance that the data being audited is complete, consistent, and accurate over its life cycle. For this purpose, the inspected site is expected to provide documentation needed to reconstruct the study events and verify the results. Data quality provides assurance that data were collected in accordance with protocols, SOPs, and applicable standards of research. OSIS reliability recommendation depends on evidence collected during inspections, affirming data integrity and quality. The next step in the process after inspection is to reporting the findings. After an inspection is completed, the details along with the inspectional proceedings and findings are documented by writing an Establishment Inspection Report or EIR. It provides the basis for the determination of regulatory and scientific impact of the findings. The reporting process involves multiple steps. First, the participating investigator or investigators write an Establishment Inspection Report or EIR and attach the appropriate exhibits and documents that support inspectional findings. The EIR package is then submitted to the center for review and final classification. The center reviewer thoroughly reviews the EIR and writes another report called as the EIR review report. It is basically an overview of the relevant inspectional details, objectionable findings, exhibits, discussion items, and firm's written response to Form FDA 483 if applicable. The final classification of NAI, VAI, and OAI is also given at this point. The final and the last step in the process after EIR and EIR report review writing is support. Support is a critical step in the process. In this final step, after thoroughly reviewing and evaluating the inspectional findings, OSIS provides recommendations to the appropriate CDER division about study integrity and quality. This information is one of the key pieces of information CDER uses in making a regulatory and approval decision for a drug application. OSIS may consult and collaborate with other offices, such as Office of Scientific Investigation, OSI, or for regulatory actions and enforcement. OSIS also collaborates with international regulatory agencies by sharing data and conducting joint inspections. Here is a summary of what OSIS does. Assigns and conducts inspections in collaborations with Office of Regulatory Affairs to ensure studies submitted to FDA were conducted properly and data are reliable. Writes and reviews establishment inspection reports and determine regulatory and scientific impact. Makes recommendations to CDER review divisions to support regulatory review and approval decisions. Makes surveillance evaluations of regulated entities conducting BA, BE, and GLP studies, and investigates allegations and complaints of regulatory non-compliance. In conclusion, OSIS applies multi-pronged approaches to ensure reliability of data submitted to the FDA as part of bioavailability, bioequivalence, nonclinical GLP, and animal rule studies in collaboration with the Office of Regulatory Affairs and the Office of Scientific Investigations. Please type your comments or queries in the chat box. I will address them during the Q&A at the end of the first session. However, I have a couple of course questions for you. There are two questions for this presentation. Question number 1, which of the following inspections are covered under OSIS's purview? A, inspection of facilities that conduct in vivo BABE studies, clinical portion. B, inspection of facilities that conduct in vivo BABE studies, the analytical portion. Inspection of non-clinical labs conducting animal rule-specific studies, D, all of the above, and E, A and B only. Question number 2, compliance programs serve as a guide for conducting activities to evaluate industry compliance with FD&C Act and other applicable laws. The potential answers are A, yes, or B, no. The correct answer to question number 1 is D, all of the above. And the answer to question number 2 is A, yes.
Good afternoon, everybody. I am one of the members of the Collaboration, Risk Evaluation, and Surveillance team, also known as CREST, within the Office of Study Integrity and Surveillance, also known as OSIS, within CDER. Today, I'll be giving you an overview of the CREST site selection model. My talk's learning objectives are to describe the role of CREST within the Office of Study Integrity and Surveillance, or OSIS, to list the types of studies assessed by CREST, to evaluate the risk factors CREST uses for selecting sites for inspection, to examine some of the team's recent metrics, and lastly, to identify some of the challenges and opportunities CREST faces in its day-to-day work. CREST has been tasked with conducting risk-based assessments of BE studies sites by, briefly, verifying accuracy of site information, evaluating site inspection history, assessing sites in the context of study conduct and complexity. Following these assessments, CREST recommends sites as a yes or no for inspection. Our work is done in collaboration with OSIS leadership, OSIS Office Director, Deputy Office Director, Division Directors, Deputy Division Directors, and team leaders, as well as OSIS reviewers, project management staff, and the primary review divisions within the Office of Generic Drugs and the Office of New Drugs. CREST assesses many studies submitted to the agency in support of applications. Most of the studies we assess are in vivo BE studies, but we also assess clinical endpoint studies, pharmacodynamic studies such as vasoconstriction studies, in vitro BE studies, pharmacokinetic, safety, tolerability studies, and immunogenicity, safety, and efficacy studies. Now we'll move on to the risk-based assessments. We look at a number of factors when evaluating a study site or submission. These can include the site's inspection history, whether previous inspections have been classified as no action indicated, voluntary action indicated, etc. Whether the site is a new site with no inspection history, the date and outcome of the previous inspection, the conduct dates of the previously inspected studies, the complexity of the studies, any unique aspects of the study conduct like a unique type of assay, and we may also look at factors identified in a for-cause request or information received from other regulatory bodies. When we are assessing a study site, we may ask ourselves some of the following questions. Do we need to clarify any discrepancies in the site address? We don't want to send anyone to a closed site or one that doesn't exist or the wrong site. Has there been a change in management? Are there known adverse events associated with the drug? Are the previously inspected studies relevant to the current studies? For example, when evaluating a clinical endpoint study, we may not be able to rely on inspection history for in vitro studies. Were there an unusually high number of discontinued subjects? For analytical sites, were there a lot of repeat analyses? Do we know of any issues with the analyte? The last few slides contained a few examples of what we consider during an assessment. Overall, there are over 30 factors that we apply to both clinical and analytical sites. Generally, risk assessments are qualitative in nature, and we are looking to incorporate a quantitative approach, OSIS and CREST evaluate the risk factors used on a regular basis, and we assess every submission, every pivotal study, and every relevant site, which means we have a lot of work. And now we move on to look at some of the workload we've handled recently.
In the previous calendar year, CREST assessed over 1,200 submissions, including ANDA submissions and NDA, BLA, IND, and clinical endpoint ANDA submissions. In total, we assessed over 2,300 sites.
As we handle this workload, we are also faced with a few challenges and opportunities. We need to manage and maintain the accuracy of information of a large international site inventory. We need to provide comprehensive risk-based site assessments taking into consideration a multitude of factors. We need to work efficiently in order to provide timely assessments to ensure user fee timelines are met. And with that, we will move on to a few challenge questions to test your knowledge on what I've discussed. Question number 1. Who is responsible for providing a risk-based recommendation to inspect a site that conducts in vivo BE studies? A. Project Management Team, PMT. B. The Collaboration, Risk Evaluation, and Surveillance Team, CREST. Integrity Services. Or D. OSIS Division Directors. The answer is B, the Collaboration, Risk Evaluation, and Surveillance Team, CREST. Question number 2. What is not a consideration when assessing a site? A, the date of the site's last inspection. B, the outcome of the site's last inspection. C, the site's annual revenue. D, the conduct dates of the previously inspected studies. The answer is C, the site's annual revenue. In summary, today we've described CREST's role within the Office of Study Integrity and Surveillance, or OSIS. We've listed the types of studies CREST assesses. We've evaluated some of the risk factors used in CREST's site selection model. We've examined some of CREST's accomplishments in the last year, and we've identified a few of the challenges CREST faces in its daily work. If you have any questions, please submit them to the moderators and we will try to address some of them during the Q&A session. Thank you very much for your attention.
This presentation title is Analytical Inspections for Bioavailability, Bioequivalence Studies. I am Li Hong Ye. I am a chemist in the Division of New Drug Study Integrity in Office of Study Integrity and Surveillance. We are part of the OTS, CDER, FDA. Disclaimer: the contents of this presentation are of author and should not be construed to represent views or policies of the U.S. Food and Drug Administration. Learning objectives. I have 4 areas to discuss. To understand OSIS conduct of analytical inspections for bioavailability, bioequivalence, in short, it is BA/BE studies. I would like to describe analytical inspections, including on-site inspection, as well as remote regulatory assessment, in short, RRA. I will share some common observations, and I will provide closing remarks. Drug inspections under FDA Bioresearch Monitoring Program, BIMO. It includes clinical investigators, sponsors, contract research organizations, institutional review board, risk evaluation and mitigation strategy, post-market adverse drug event, and good laboratory practice, GLP, and animal rules, AR. And the last item is bioavailability, bioequivalence, BA/BE. We will focus on the BA and BE part. Where is OSIS? OSIS composed of two divisions, Division of the New Drug Study Integrity and Division of Generic Drug Study Integrity. We are under the super office of Office of Translational Science, OTS. It is part of the Center for Drug Evaluation and Research, CDER, and we are part of the US FDA. Another office, Office of Regulatory Affairs, ORA, also conduct inspections for clinical part. For BA/BE Bioresearch Monitoring BIMO program, the objectives of the In Vivo BA/BE Bioresearch Monitoring program include to ensure the protection of the rights, safety, and welfare of human subjects participating in studies, to ensure the quality, integrity, and validity of clinical, analytical, and statistical data from BA/BE studies, to ensure compliance with applicable FDA regulations, and to identify significant deviations. What is OSIS assignment workflow? First, OSIS selects the sites for inspection. Then, OSIS sends inspection assignments to OSIS staff. OSIS then conducts analytical inspection. OSIS prepares the Establishment Inspection Report, EIR. OSIS then reviews reports and exhibits and prepare OSIS review memo. OSIS would also issue closeout letter. Let's take a closer look of analytical inspections. One way OSIS conduct the inspections is through remote virtual regulatory assessment. Following is the overview of the process. OSIS implemented a remote evaluation tool in June 2020. For remote regulatory assessment, RRA, FDA has a draft guidance for industry on conducting remote regulatory assessments published in July 2022. RRA allows OSIS to continue to support the generic, biosimilar, and new drug review programs through oversight of bioavailability, bioequivalence, GLP, and animal rule studies during the pandemic. For the on-site inspections, I will spend more time to talk about inspectional focus and its expectations. It includes method validations, which such as stability, methodology, documentation, analysis of study samples. We can reference to M10, Bioanalytical Method Validation and Study Sample Analysis Guidance, which was published in November 2022. For the OSIS inspection, I will focus on the analysis of study samples, which include system suitability, run acceptance, re-injection of study samples, internal standards, variability, the trend, the PK anomalies, and reassay. Also, it includes the incurred sample reproducibility. I will talk in more details of OSIS audit in the analysis of study samples. First is regarding system suitability, SS. Independent of study sample analysis, evaluated prior to analytical sample run. It is also evaluated prior to resuming a sample analysis run after any instrument malfunction. For run acceptance, we audit at least 3 QC levels in duplicate and LLOQ. The run meets acceptance criteria such as 75% CCs at 6 levels, at each level within 15% of nominal, and also 20% LLOQ, as mentioned in M10 guidance. We also look at the reinjection of study samples. If it is justified, if it is SOP-driven, sample covered by validated processed sample stability, and all reinjected samples reported in study report. We also look at internal standard variability, ISR, and including the drift and trend. SOP to determine run rejection based on variability or drift in IS response. IS variability limits pre-specified in SOP, QCs, and samples with IS variability outside limit reanalyzed. We also look at PK anomalies, PK samples reassay governed by SOP, potential reason investigated, sample repeat request documented with reason, anomalies and final concentrations reported with reason for selecting final concentration. We also look at incurred sample reproducibility, ISR. Samples representing analyte Cmax and elimination half-life meet predefined acceptance criteria such as 67% samples values within 20% of original sample values. Procedures predefined for investigation of ISR failure. When OSIS conduct inspection, we ask the question, does the documentation allow us to reconstruct the study activities? I would like to share some common analytical observations during our analytical BA/BE inspections, such as method not fully validated, inadequate stability data to cover sample storage and handling, incomplete documentation to allow reconstruction of study activities, reinjection of analytical runs that meet acceptance criteria, inconsistent run acceptance, data security, including inappropriate access privileges. In summary, OSIS supports FDA mission by ensuring data supporting regulatory decisions are reliable. OSIS conducts on-site inspection of analytical sites for surveillance purpose. Remote Regulatory Assessment, RRA, is a tool that OSIS used during COVID-19 pandemic and will continue to use in assisting FDA in accomplishing its mission to protect public health. Data submitted to FDA should be complete, accurate, and reliable to ensure safety, efficacy, and quality of drug products. For challenge question 1, OSIS conducts analytical site evaluations to ensure data integrity by on-site inspection only, true or false. Challenge question number 2. Which method OSIS uses to conduct BA/BE site evaluation? On-site inspection, remote regulatory assessment, both, or none of the above? Attention and participation. Any questions?
Right. Thank you to our presenters for their excellent presentations and insights. We'll now begin our first question and answer panel. Please note, joining this panel in place of Dr. Gabriel Davila will be Dr. Clint Mitchell, lead pharmacokineticist within the Office of Study Integrity and Surveillance regarding the subject of the second presentation. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. So we have a few questions coming in, and we'll start with a first question here for Dr. Mirza. Dr. Mirza, we noticed on your slide number 12, that's the site assessments by submission type, that you didn't have a part on your pie chart for INDs, only NDAs. Does OSIS also inspect studies from INDs?
Hi, that's a good question. This is Taseen Mirza. I did put a pie chart because in the interest of time, we get involved in any BAB study, whether it's in IND phase or NDA phase. I just didn't put it in the pie chart because of time constraints. So yes, we are involved in IND as well. If it's a BAB study or a clinical study, part of it is taken care of by ORA with our help. But for the analytical part, we do the inspection ourselves.
Thank you so much for that response. We have another question for you, Dr. Mirza. Is your work also under the scope of Compliance Guidance 7348.810 for sponsors?
Yeah, this is for sponsors. The best way to answer that is, it is part of BIMO, BIMO inspection, and it is performed by ORA, and our sister office, OSI, Office of Scientific Investigations, gets involved. Our involvement is minimal at best, but yes, it's part of BIMO, and it is performed mainly by our sister office, Office of Scientific Investigations, so OSI.
All right, thank you for that response. Oh, sorry, was there more to add?
No.
Okay, thank you. We appreciate that response as well. Now our next question has come in for Dr. Clint Mitchell. Regarding CREST assessments, are the Office of New Drug or O&D review divisions typically consulted or involved in site selection? Under what circumstances might CREST reach out to a review division for more information?
Sure, thank you for that. That's a good question. Actually, for NDA, BLA, and many IND applications, the Office of New Drugs, or O&D, their primary review divisions actually send us the specific sites that are part of a particular study they are reviewing, and they will request that these sites be evaluated by OSIS to see if an inspection is needed. So you could say that O&D is very involved in the site selection for these types of applications. For the second part of the question, I would say that there's probably many different reasons why CREST might touch base with O&D to communicate on the sites that they've requested for inspection. A lot of the times it's because we have additional information regarding some of these sites that they might not be privy to, or maybe there might be other sites involved in the application that could be problematic that we would prefer to inspect other than the ones that they initially send us. So sometimes there is quite a bit of back and forth between us and O&D just to collaborate on the best path forward. So thank you for that question.
All right, thank you so much for that response as well. Another question for you, Dr. Mitchell. Could you please share the site selection model for surveillance?
Thank you. Yeah, that's probably the million-dollar question, right? What are the 30 or so risk factors that Dr. Davila outlined? We get that question a lot, but we do not publicly share those risk factors that OSIS utilizes when determining whether or not an inspection is needed. However, I would say that, and I see some other questions along the same line have come up. If your firm or CRO conducts the clinical or analytical portions of BABE studies that are submitted to the FDA, it's currently our goal to inspect your site every 2 years, plus or minus, 6 months or so. But that inspection could be an on-site inspection every 2 years, or it may also entail a remote type of audit, like Paul mentioned, the remote regulatory assessments, or RRA. So thank you for that question as well.
And thank you again for that response. We now have a question for Dr. Ye. Are PK and immunogenicity studies included in the scope of CP for In Vivo BA/BE studies analytical?
Thank you for the question. The answer is yes. For OSIS, we do conduct inspection or conduct the RA for all the studies related to the PK and the immunogenicity studies, which is under our inspection scope. Thank you.
Thank you so much. Now we'll go to a question again for Dr. Mirza. What is a remote regulatory assessment, or RRA?
Yeah, it's a very good question, and it's asked frequently. A remote regulatory assessment is assessment of data and the site inspection remotely from an inspector's office or any other alternate site. RRA was developed out of a need because during COVID, we couldn't travel, but the work had to be going on. So for that reason, we had to quickly develop a process by which we can inspect a site remotely, authenticate data that's submitted to FDA, and ensure patients were not harmed, and so on and so forth. So it is a elaborate process that was developed during COVID for inspecting sites remotely. So there follows specific steps. Initially contacting the site to see if they're available, make sure the technology is available on the site, so we can use video audio facilities properly. And sometimes during emergency, even staff is not available on the other side. We have to ensure that. And once we do that, we also have to ensure that we can share documents. Documents can be large. So we found a way to dump documents in a neutral site from where we can pick it up and inspect. So basically, it's a process by which we can inspect site remotely. Hence, it's called as remote regulatory assessment. Thank you.
Thank you. Thank you for that information. On that note, another question for you. Will OSIS continue to use RRA in the future?
That's a good one. We look at RRA as a tool, an additional tool in our toolbox, meaning that we can perform RRA by itself. We can do inspections, on-site inspections, or we can use a hybrid mode. For example, before we go to a site, we may be able to do some things remotely. For example, check the source data by contacting or conduct meetings with staff before going to the site. Or it is possible, once on the site, we decide that we need a little bit more time, can come back and complete the inspection by a remote process. So going forward, it's very hard to say when and where it will be used, but it's an additional tool we have, and time will tell whether we'll use it as is or as a hybrid mode with inspections. If you were to ask me, I think it'll be used in all different forms, as a standalone as well as hybrid mode. Thank you.
All right, thank you so much for that response. We have one more question for you, Dr. Mirza. What is a compliance program or CP?
Compliance program. That is compliance program is a document, detailed document, which describes how an inspection should be conducted. It uses a standard, higher standards of inspection practices, and it also ensures that the inspection is performed under the purview of the authority given to us, that is the different regulations. So a CP is like a cookbook for a recipe book for inspectors to follow when they go to a site. What CPs do is standardize the inspection process. In other words, different inspectors cannot be using drastically different approaches to inspection. It gives some sort of standard process for inspections once they are at the site. So a compliance program is one of the most important documents an inspector has for conducting inspections. Thank you.
All right, thank you so much for that information. We have now a question that came in for Dr. Mitchell. Does OSIS also inspect phase studies like first in human studies?
Sure, great question. Yes, is the direct answer. Yes, we've audited many phase 1, 2, 3A, 3B studies like that, typically safety, efficacy, tolerability studies, as well as immunogenicity studies, wherever those might fall on that phase 1, 2, and 3 spectrum. So yes, thank you.
All right, thank you so much for that response. We'll go to another question for you, Dr. Mitchell. My firm was just audited by OSIS for a bioanalytical study. When will FDA/OSIS inspect my facility again?
Another good question, one we typically receive quite often as well. Thank you. Again, in general, our goal is to inspect a given facility every 2 years. However, as you all may know, concerns might arise during the review process that might cause FDA to conduct an inspection sooner than that 2-year interval. So 2 years is the goal. Thank you.
Another question for you. Does OSIS inspect sites in the Middle East?
Yes, great question. And the answer is yes. We've audited many firms in the Middle East, sites in Jordan, Israel, UAE come to mind off the top of my head. But yes, we definitely do. And we've done both on-site inspections as well as RRAs, remote regulatory assessments. So we've done both in that region of the world. Thank you.
All right. Thanks. And kind of related to that, does OSIS inspect foreign CROs more or less often than CROs located in the United States?
Another good question. No, OSIS applies our risk criteria equally to CROs, really regardless of where they are located throughout the world. Thank you.
All right, thank you so much for your responses. Now we'll move on to some questions for Dr. Ye. Does OSIS prefer on-site inspection or remote regulatory assessment?
This is an excellent question. Both on-site inspection and the RRA helps OSIS to evaluate the study conduct and the site operation. Both are important tools for OSIS to use, so there's no preference between these two inspection tools. Thank you.
Right, thank you for that information. Another question for you. When conducting an on-site inspection, does OSIS expect the site to provide a printout of the audit trails for review?
That's a great question. OSIS want to reconstruct what happened during the study conduct. Thus, we can review the audit trails in the system for the sample analysis. We might also choose the specific time period for review. So it is not necessary to print out PDF files for review when we are conducting the inspection. Thank you.
Thank you so much for that response. We had another question that came in for you. Is it recommended to submit a system suitability with every sequence of analytical run, or only one-time system suitability is required for each day?
Thank you for the great question. I think it all depends on how your SOP define how you conduct the system suitability and how you design the system suitability to be used for the subject sample analysis. The purpose for the system suitability to ensure that the equipment used for the subject sample analysis is properly performed and functioning as expected. So the frequency to conduct the system suitability, or how do you conduct as a part of the total sample analysis program, all depends on your own SOP. Thank you.
All right, thanks so much, Dr. Ye. We have actually one more question for you. For RRA, does the site need to upload all documents before OSIS can initiate the opening meeting to conduct the remote regulatory assessment?
This is an excellent question. We note that RA is voluntary, so OSIS staff would work closely with the site about the requested document and the expectations of the opening meeting. We can also request documents during the RA. Thus, it is not a requirement that all documents to be uploaded before the beginning of the opening meeting of the RA. Thank you.
All right, thank you so much for that response. And that looks like all the questions we have time for now. Thank you again to all our presenters for their excellent information. And so now we'll take a short break to resume in 15 minutes. And welcome back from the break. Hope everyone was able to take a moment to refresh before our next sessions. We'll now hear from our next set of presenters. First, we'll hear from Dr. Monica Javidnia, a staff fellow within the Division of Generic Drug Study Integrity, or DGDSI, in the Office of Surveillance and, the Office of Study Integrity and Surveillance. And her presentation will be followed by Dr. Mark Seaton, a senior pharmacokineticist, also within the division, also within DNDSI within OSIS. Now, please join me in welcoming Dr. Javidnia.
Thank you for having me, and thank you to everyone for joining us for this webinar. I'm Monica Javidnia, a staff fellow in the Division of Generic Drug Study Integrity on the clinical team, and I'll be giving an overview of clinical inspections for bioavailability and bioequivalence studies. As a disclaimer, this presentation reflects my views and should not be construed to represent FDA's views or policies. The learning objectives for today's presentation are to understand OSIS's collaboration with the Office of Regulatory Affairs, ORA, on clinical inspections for BA/BE studies, describe clinical inspections, and to identify common issues found during inspections with a couple case studies. Let's start with an overview of BA/BE clinical work in OSIS. Clinical inspections through OSIS fall under FDA's Bioresearch Monitoring, or BIMO, Compliance Program 7348.003 In Vivo Bioavailability, Bioequivalence Studies, Clinical, as opposed to 7348.811, Clinical Investigators and Sponsor Investigators. I've included a link in the slides. This is a really helpful document that details the program objectives, inspections, and a lot more. So if you are not familiar with this document, I highly recommend checking it out. OSIS is an office under CDER's Office of Translational Sciences. Within OSIS, we have two divisions, the Division of New Drug Study Integrity, DNDSI, and the Division of Generic Drug Study Integrity, DGDSI, where I am. Both divisions work on both new and generic drug applications, as well as BLAs and INDs, and both divisions have two analytical teams. However, DNDSI has a GLP team, and our clinical team is within DGDSI. We also work closely with the OSIS Immediate Office, specifically the Project Management Team, Collaboration Risk Evaluation and Surveillance Team, or CREST, and the Associate Director of Clinical Policy. OSIS works with ORA before, during, and after inspections or remote regulatory assessments, RRAs, of clinical sites. Here's our general workflow. I won't go into much detail about CREST because I know they gave a great presentation earlier on all the amazing work they do, but very briefly, CREST will review applications that come to us, the studies, the associated sites, complaints, other concerns if applicable, and then select sites for inspections. The studies can be PK endpoint, clinical, immunogenicity, analytical, including in vitro bioequivalence studies, but I'll be focusing on the clinical side for this talk. OSIS then sends the inspection assignment to ORA. The ORA investigator will conduct the inspection, or RRA, and then prepare a report of their findings and gather all the exhibits. So if this is an inspection, the report will be in the form of an establishment inspection report, and if it's an RRA, it'll be an RRA memorandum. The reports have very similar content, but RRAs are not inspections and as such will not have an EIR. The OSIS reviewer will then review ORA's report and any associated exhibits. If there was an FDA Form 43 issued, we'll review that, as well as any response from the site. Afterward, the OSIS reviewer prepares an OSIS review. This document serves to summarize the inspection or RRA and provide recommendations to the applicable review divisions regarding study conduct, subject safety, data reliability, and regulatory compliance. OSIS will also issue the closeout letter to the person identified as the most responsible person at the site. This is a very high-level letter, basically recapping that there was an inspection or RRA at their site and the goals of the bioresearch monitoring program. However, if there were more serious findings or regulatory violations, the letter would include additional details and likely be issued from CDER's Office of Compliance. Now that I've given an overview of BABE clinical work in OSIS, let's dive into clinical inspections. FDA's Bioresearch Monitoring Program ensures that the information submitted to FDA are scientifically valid and reliable, and that the rights, welfare, and safety of human subjects have been protected. To support that goal, FDA conducts inspections to verify the reliability, integrity, and compliance of clinical and non-clinical research being reviewed in support of pending applications. After receiving the assignment from OSIS, ORA will determine whether the site should have an inspection or an RRA and identify the investigator or investigators who will be conducting the inspection or RRA. For this talk, I'll focus on inspections, but briefly, RRAs are remote evaluations. As I mentioned earlier, they are not inspections, but they do have a similar format and coverage that an inspection would have. They're another tool that aids us in providing recommendations to the appropriate review divisions. For inspections, ORA may arrive unannounced or announced. They'll present their credentials. For domestic sites, ORA will also issue FDA Form 482, which is a notice of inspection to the most responsible person at the site. So, what are some things that the ORA investigators would be looking at when they're on site? This is not an exhaustive list, and some aspects may or may not be applicable to all study types. There will typically be a tour of the facilities. This helps ORA evaluate the site, the workflow for study activities, spaces subjects would be in, like dining areas and bedrooms. If the site has a floor plan, ORA may obtain a copy of that. ORA will also interview personnel involved with the study or studies. If the site has an organization chart, they may be asked to provide a copy of that too. They'll review the protocol and any amendments, as well as the Institutional Review Board or Independent Ethics Committee approvals. As part of that, definitely the protocol deviations. The informed consent forms, including ensuring subjects were properly consented, they were consented on the correct forms, and reviewing the language in the forms themselves. Of course, source documentation, case report forms, and/or electronic case report forms, ECRFs. Do the source records match the CRFs? If the study used paper and ECRFs, do the data match? As part of that, some components reviewed are subject screening and eligibility, not only at the start of the study, but throughout. If there were any food or water restrictions, like for a fasted study or a food effect study, they'll review those records, PK sampling times, laboratory test results, and how those test results were reviewed. Also, adverse events and serious adverse events, how they were reported, how they were followed up, and what the resolutions were. Other items reviewed are randomization and blinding, including procedures for ensuring the blind or double blind was maintained. Records related to the investigational products, such as drug administration, shipping and accountability records. If applicable to the study that was conducted, reserve samples will also be reviewed. I've included 2 links to the Code of Federal Regulations pertaining to reserve samples. If there are any relevant standard operating procedures, they will be reviewed to ensure that they were followed and that they're sound, whether that's scientifically or compliance-wise. For equipment like freezers where PK samples were stored or spaces where drugs were stored, temperature monitoring records will be reviewed in addition to calibration and maintenance records for critical equipment. Within this, other records or documents will also be reviewed. For example, if there was a temperature excursion, is there documentation of it? How long was the excursion? What were the maximum temperatures reached? If the samples were removed, is there documentation of that? Was there any protocol deviation filed? If there's an SOP on temperature monitoring, was it followed? So while I listed these components as bullets, there's a lot of connection between them. I again mentioned that this was not an exhaustive list. There's so much more that ORA will review when they're conducting an inspection: sample processing times, training records associated with the study, delegation of responsibilities logs. Far too much to list, but I hope this and the preceding slide gave a good overview. An inspection typically takes 5 days. At the end of an inspection, the ORA investigator holds a closeout meeting. If there are objectionable conditions identified during the inspection, the ORA investigator will share them with the site at the closeout meeting, and the site has an opportunity to respond and ask questions. There are categories of objectionable conditions. The first is an observation. These are conveyed to the site verbally and in writing by issuing FDA Form 483, inspectional observations, to the most responsible person at the site. The other is a discussion item. These are verbally discussed at closeout. The site will have 15 business days to provide a response if they choose to. The OR investigator will prepare an establishment or inspection report. Sites will get a copy of this report. And as I mentioned earlier during the assignment workflow, we at OSIS review the EIR, the exhibits, and if there's a 483, the site's response. We'll prepare the OSIS review, providing our recommendations to the review divisions. We will also issue the final classification. Inspections are classified as either NAI, no action indicated, VAI, voluntary action indicated, or OAI, official action indicated. I've included a link to the FDA website that goes into further detail about the classifications. Now that I've described the inspection component, let's get into a couple common issues that we see and a case example for each. Two of the most common issues are with protocol adherence and with record keeping. Just as an aside, if you're interested in FDA inspection data, this website I have linked in the slides has all sorts of dashboards with information like number of domestic and foreign inspections conducted by fiscal year, inspection classifications, classifications by product type such as tobacco or drugs, top 10 citations, and more. This dashboard is for all FDA inspections. It's not specific to OSIS's clinical BIMO work, but it still has a lot of interesting information. So let's start with protocol adherence. As I shared earlier, ORA investigators will review the protocol and amendments as part of their inspection. This helps ORA ensure the protocol was followed when they review other records, that subjects were eligible to participate in the study, amendments were approved before being implemented, visits occurred according to the schedule, samples were collected and processed appropriately, protocol deviations were properly filed, and a lot more. Case study number 1 is for a food effects study. The protocol said that the study drug should be administered 30 minutes after the start of the meal. While ORA was reviewing records, they found that 14 of the 15 subjects in the study received the study drug before the 30 minutes had elapsed since the start of their meal. In our OSIS review, we recommended that the review division determine the impact of the protocol deviation on PK data. Record keeping is also a really important component of study conduct, and this is a very broad category, with issues including, but not limited to, failure to retain records, discrepancies between two records, for example, the paper case report form not matching the electronic case report form, illegible records, so we may not be able to tell who performed a task or what notes are on an adverse event sheet. Proper record keeping is important for many reasons, including ensuring data integrity and allowing for the ability to reconstruct and follow a study. For our second case study, this had to do with the freezer records at a clinical site. ORA found multiple issues, some of which I've included on this slide. There were several instances of more samples being stored or retrieved on the freezer log than were actually collected from the study subjects as part of the study. So say 500 samples were logged, but only 400 were collected during the study. Sample aliquots were logged for storage on a certain freezer shelf, but later when they were retrieved, the log listed them as being on another shelf, and there was no record of any samples being moved from one shelf to the other between the initial storage and the retrieval. Another issue identified was no record of samples being returned after retrieval. This is another place where record keeping is important to know whether samples were returned, shipped elsewhere for analysis, or destroyed after storage for a period of time. Overall, these highlighted issues related to sample intactness and the inability to trace the samples. Again, these were just some of the problems identified, and it was a systemic issue rather than isolated to a specific study, which we also conveyed to the review division. All right, let's do a couple challenge questions, and then I'll share some resources you might find helpful. Challenge question number 1, which of the following statements is not true? OSIS's clinical BIMO program falls under Compliance Program 7348.003, In Vivo BA/BE Studies Clinical. ORA provides the final inspection classification. OSIS sends the inspection assignment memorandum to ORA. Or D, ORA writes the establishment inspection report following an inspection. I'll give you 10 seconds here. All right, the answer is B. OSIS will provide the final inspection classification. Challenge question number 2, which of the following study components could FDA inspect? Adverse events, drug accountability, IRB or IEC approvals, or D, all of the above. I'll give another 10 seconds here. The answer is D, all of the above. Here are some regulations relating to good clinical practice and clinical trials. I shared a couple links earlier to information about reserve samples. These are under 21 CFR Part 320. And here is some more helpful information. FDA has a lot of great resources like trainings, seminars, continuing education, of course these SBIA webinars. I highly recommend the FDA Clinical Investigator Training course held annually. Also, there is a webpage for good clinical practice inquiries where you can ask questions and also see responses to previous questions. In summary, OSIS collaborates with ORA for clinical bioavailability and bioequivalence inspections. ORA conducts the inspections, then OSIS provides recommendations to the review divisions based on evaluation of the inspectional findings. I just have a couple closing thoughts. When I was interviewing for grad school, I was asked what I want to do, and I said my goal is to get safe and efficacious drugs to the people who need them. Whether I was at the bench or in my current position with OSIS, it's still my goal, and as part of that, I really do view us all as being on the same team. I hope you found my talk and in general these events FDA and SBIA hold to be helpful, and that you feel prepared if ORA shows up at your site one day. Unfortunately, I'll be unavailable for questions, but Dr. Julia Cho will be answering questions related to this talk in the upcoming Q&A panel session. I would like to thank you all for your time and attention, and I'll turn it over to my colleague Mark for his presentation on our GLP program.
Thanks, Monica, and good morning, everyone. My name is Mark Seaton, and I'm part of the GLP team in the OSIS Division of New Drug Study Integrity. I'm happy to give you a short introduction to FDA Good Laboratory Practice regulations. Before I start, the opinions and information in this presentation are those of the author and do not necessarily represent the views and/or policies of the U.S. Food and Drug Administration. Here's the plan for the next 20 minutes or so. First, I'll describe the history and scope of good laboratory practice regulations. By the way, good laboratory practice takes way too long to say, so I'm going to abbreviate that as GLP and probably say it a few hundred times. Then I'll describe how OSIS is involved with GLPs and a bit more broadly what the FDA does with respect to GLPs. For those of you who like an outline, here you go. In case you're wondering, CFR here is Code of Federal Regulations. Don't worry, that won't be on the quiz, but you can search the internet for 21 CFR Part 58, and I'm pretty sure you'll find a link to the GLP regulations. So as you see, we'll go over the regulations a little bit. I'm going to talk about the role of OSIS with GLP, and then I'll end up with an overview of the GLP inspection program. As I suggested, GLP regulations are listed in 21 CFR, and GLP is a quality system used for non-clinical safety studies to support investigational and marketing applications for products regulated by FDA. In FDA CDER, non-clinical toxicology and safety pharmacology studies are the first step in understanding safety concerns that drug developers and the FDA should be aware of and are essential to support the safety of human clinical trials. The GLP regulations are used to assure that tox studies are appropriately conducted and documented to generate reliable data. So why do we need GLPs? Well, before GLP regulations were established, FDA performed just a limited number of inspections of labs that performed non-clinical safety studies. Those inspections would be at the request of FDA review divisions, when scientists in those divisions who were reviewing the data submitted by drug developers found issues or had concerns. There, at the time, were no published regulations to guide the conduct of either the non-clinical studies or the FDA inspections. Most labs were never inspected, but unfortunately, at the ones that were inspected, FDA found many cases of poor lab practice and fraudulent activities. So in the early 1970s, FDA started a series of inspections at 40 labs just to sort of get a lay of the land. That effort revealed that in many cases, experiments were carelessly executed and or inaccurately analyzed and reported. Often the technicians and scientists working on the studies didn't have appropriate education and training, and data generated during a study wasn't reviewed to verify accuracy and completeness. Looking at the data that was reported in terms of accuracy. This slide provides some good quiz material, but suffice it to say that the FDA's Bioresearch Monitoring Program, referred to as BIMO, was established in 1977 with the mission of verifying the data submitted in support of investigational and marketing applications, and to provide oversight of the conduct of non-clinical safety studies, as well as clinical trials using regulated products. Now, this text is taken directly from the regulations. I want to emphasize here that GLPs are meant to assure the quality and integrity of the data, but not the quality of the study design itself. Along those same lines, here's some other examples of what GLPs don't do. This is a list of studies that did not fall under the scope of GLPs. You see exploratory work and clinical trials on the list. This is just a quick note about non-clinical safety studies conducted at foreign sites. Many of those studies state that they are following GLP principles published by the Organization for Economic Cooperation and Development, or OECD. The OECD GLP principles were developed in 1978 and were based on the FDA GLP regulations. The OECD GLP principles were revised in 1997, but the primary objective here is similar to FDA, to ensure the generation of high quality and reliable test data related to the safety of industrial chemical substances. Since there's enough similarity between FDA Part 58 and the OECD GLP principles to allow compliance with both, if foreign sites follow OECD principles, it's likely that they are compliant with Part 58. Now, for those of you following along on the outline, we're moving to item 2, role of OSIS with GLP. I'll briefly describe the roles and responsibilities of those of us on the OSIS GLP team. So the mission of the OSIS scientists on the GLP team is to support the CDER pharmacology toxicology review process by verifying data quality and integrity. For the most part, we do that through surveillance inspections to assess non-clinical laboratories' general ability to conduct GLP-compliant studies. But we also issue directed inspection assignments based on PharmTox reviewer concerns. Also, we occasionally receive complaints from the public about regulated firms, and we investigate those as well. Now, more specifically, the GLP team does site selection for GLP inspections and selects the specific studies to be audited during those inspections. We also prepare inspection assignment memos, and in some cases, we participate on those inspections. When we do, we help prepare the inspection report. In all cases, we perform a compliance review for each inspection based on the inspection report. And then based on that compliance review, we might take regulatory action if it's appropriate. That would come in the form generally of correspondence to firms requesting improvement. And finally, we'll make recommendations to the CDER review divisions, specifically farm tox reviewers, in order to support the regulatory review and approval decision. In this last section of my talk, I'll describe the GLP inspection program and go a little bit into what you might expect on an FDA inspection. By now, you've heard that the OSIS GLP team is responsible for assigning, reviewing, and in some cases even participating on GLP inspections to verify quality and integrity of non-clinical study data. We select for inspection studies that are pivotal to the review division's safety decision, that is, studies used to protect human safety in clinical trials. We want to make sure those data in particular are reliable to support the review decisions in IND, NDA, ANDA, and BLA applications. And just in case you were wondering, the cost of these inspections is covered by user fees paid by the companies that submit those applications. The individual labs themselves do not cover the cost of the inspection. And FDA does not issue FDA compliance certificates. Because of limited personnel and limited resources, FDA and OSIS in particular can't cover all firms and audit all studies. You might find it surprising that the OSIS GLP team prioritizes inspections of animal rule efficacy studies. That would be surprising because animal rule studies don't fall under the scope of GLPs. But the efficacy studies performed under the animal rule replace human efficacy trials, so that data integrity and reliability is absolutely critical for review division approval decisions. Directed, also called for-cause inspections, are next in terms of priority for the OSIS GLP team, and we use those to address specific review division concerns. We'll confirm data integrity and reliability of pivotal studies. In some cases, directed inspection assignments are issued to follow up on complaints. Those are often submitted anonymously and might be related to data falsification or lack of GLP compliance. Generally, a member of the GLP team will participate on directed inspections. Surveillance inspections may be at the bottom of the priority list, but they do make up the majority of the inspections that the OSIS team manages each year. As stated here on the slide, these inspections are periodic, generally every 2 to 3 years, depending on how many studies a lab submits to the agency each year. OSIS doesn't tend to participate on most surveillance inspections, mainly due to the greater number, but we might be out there. And these inspections are meant to be an overall assessment of the firm's compliance with GLP regulations. So a bit more about surveillance inspections, since they do make up the bulk of our program's efforts. The number of surveillance inspections is set at the beginning of each fiscal year. And as I said, we visit a lab every 2 to 3 years. Now, that might be a bit longer if the lab doesn't submit many studies each year. It could be 4, maybe even 5 years. So now, because there might be some interest, I'd like to walk quickly through an overview of how an inspection is conducted. At the start of an inspection, there will typically be a documentation request, and that would include organization charts, a floor plan, a master schedule, which is basically a list of studies that the lab has and is performing, an index of standard operating procedures, abbreviated SOPs, an equipment index, so we know what type of equipment the lab has and is using, and we'd like to see maintenance and calibration schedules for those equipment. We'll ask for specific study director and study personnel training files, and we want to see pest control records. So during the inspection, the inspector or the inspection team will conduct a facility tour, and usually along that tour we'll meet different technicians involved in study activities, and we'll interview them, make sure they understand their roles and responsibilities. After the facility tour, which could take one day, or if it's a large facility, more than a day, the inspector or the team will review lots and lots of records. Those are going to include study-specific documents, study data, study records, but also facility and equipment records, maybe temperature monitoring, humidity monitoring in different rooms and in refrigerators and freezers. We're going to look at standard operating procedures, and then we're going to look at study records to make sure those SOPs were followed. We're going to look at training files, make sure people have the proper education and training. And an inspection will typically last 5 days. Maybe if it's a smaller firm, it'll be shorter. And if issues or concerns start popping up, maybe we'll be there longer. Areas of focus during inspections. It's quite a list here. I won't read it, but test and control article are important, obviously. Study director responsibilities are something that we do take seriously. Quality assurance unit is really our FDA's eyes and ears within a facility, and we'll want to make sure that they are functioning as per regulations, but also that they're getting the support that they need from test facility management. We're going to look at their SOPs, see how well they're written, how often they're reviewed, and if they're being followed. And more we're seeing electronic data capture systems being used. We want to make sure that those are used properly. They're capturing all the data, and they're capturing any changes to the data. Very important that any changes to electronic data be properly documented and a reason for the change be included in the record. And at the conclusion of the inspection, there will always be a closeout meeting. If no deficiencies are found, then no Form FDA 483 is issued. The Form FDA 483 inspectional observations is given to management and will list any deficiencies or observations found during the inspection. Some findings could be corrected during the inspection, but they would still be listed on the Form FDA 483 with a note in the inspection report that corrections were made voluntarily. And then there may be discussion items that came up during the week that didn't rise to the level of an observation or a deficiency, but will still be documented in the inspection report. Then once we're back home, if the OSIS GLP team participated in the inspection, we would be writing our portion of the EIR. That is the establishment inspection report. Otherwise, ORA, which is our FDA field office, prepares the report and submits it to us for review. That's that compliance review I mentioned earlier. We'll write that compliance review and make any necessary recommendations to the appropriate review division. Just quickly, I'll go over these Establishment Inspection Report or EIR classifications. This is the overall inspection classification. If there were no findings, no objectionable conditions were noted during the inspection, that would be labeled NAI or no action indicated. If there were some, maybe not too many and maybe not terribly significant objectionable conditions or practices, something on the level of the agency is not prepared to take or recommend any administrative or regulatory action, those would be classified VAI, voluntary action indicated. And if there are serious objectionable conditions at a lab, that inspection would be classified OAI or official action indicated, and those are cases where regulatory and or administrative actions would be recommended. So that brings me to the end of my presentation. To sum it up, valid non-clinical safety data are essential to the safety assessments for clinical trials. The GLP regulations provide the framework to ensure the quality and integrity of data from non-clinical studies. And OSIS reviewers on the GLP team work to provide assurance that the data supporting regulatory decisions are reliable. Now, before I let you go, I've mentioned a quiz several times, and so here we go. I'll give you a few seconds to ponder this one. So which of the following is under the scope of GLP regulations? Is it non-clinical PK studies? Is it human clinical studies or trials? Is it non-clinical toxicology studies or pharmacology studies for efficacy and mechanism? Give you a second. And the answer is C, non-clinical toxicology studies. All right, well, the organizers told me that question was too easy, so I had to add a second question. A bit more challenging, but I know you've got this. So, challenge question number 2. Which of the following statements is not true regarding the OSIS scientists' work with GLP? Starting with D this time. OSIS GLP inspections are performed in US only. Or is it A, OSIS scientists perform GLP inspections? OSIS scientists review inspection reports. Again, which is not true. And C, OSIS scientists prepare inspection assignments. Got your answer? Lock it in. And the answer that is not true is D. In fact, some GLP inspections are performed by the OSIS GLP team in foreign countries. And I'm going to leave you with this closing thought. Remember that GLP is a quality system that ensures the non-clinical safety studies are appropriately conducted and documented to generate reliable data. I'll thank you, and we'll move next to our Q&A session.
All right. Thank you again to our presenters for their wonderful presentations and insights. We'll now begin our next question and answer panel. As mentioned previously, joining this panel will be Dr. Julia Cho, Division Director of the Division of Generic Drug Study Integrity, that's in place of Dr. Monica Javidnia. So if you haven't had a chance to enter your question into the Q&A chat pod, please do so now, and we'll answer as many questions as time allows. And so we see some questions coming in. First questions will go to Dr. Cho. First question, is the US FDA planning more inspections for India clinical trial sites that are performing patient-based PK studies?
Okay, thank you for the question. So as was explained before, that our site selection is based on the application that comes in, and it comes with a variety of the study type, and one of them is going to be patient PK study. The way that we evaluate the sites for selection of inspection is based on the parameters that was described earlier. And we will inspect both the patient-based PK study as well as the volunteer-based healthy subject study equally based on the inspection histories and all the parameters that was talked about. So thank you for the question.
Thank you for that response. Another question for you, Dr. Cho. Could you please explain the difference between an EIR and closeout letter?
Thank you for the question. So EIR stands for the Establishment Inspection Report, and that is generated at the after the inspection is complete. When the investigator comes back to the office, they start putting all the materials that was reviewed and the inspection details, and that is what's called EIR, that describes all the inspection findings, evidences, and activity that was conducted. And once that's done and the EIR has been reviewed by the center, in case the inspection was done by the field investigator, then the recommendation is made to the review division. And when the case is completely kind of closed for the FDA, for the agency, then we call the closeout of the case. And once the case is or the inspection is ready to be closed, we also send a information and communication letter to the site, which is called closeout letter. And that closeout letter simply let the site know that we are notifying them that we are closing out the case. If there were no objection of findings found, then it would be simply stating that. If there were issues found from the inspection, then we also remind them that the corrective actions are needed, and I think that we are just kind of letting them know. So the difference between, again, EIR and then closeout letter is EIR is a report of inspection, and closeout is letter is issued to the site to notify that the case has been closed. Thank you for the question.
Thank you for the response. We have another question that came for you. Does the ORA also present a Form 482, not sure if that's supposed to be Form 83, to the facility if they are ex-US? If not, is anything presented to ex-US facilities?
Good question. Thank you for this question. So it's a Form 482, which is issued to the domestic site. And this is called issuance of inspection. Just because of the regulation that's in place, we issue 482 to only domestic sites. We do not issue it to the ex-US site. Although we want to make sure that when we say we are at the ex-US site to say that we are there from FDA, we do present our credentials to verify who we are, where we are from. That kind of proves that the inspection is official and we are from the FDA. But Form 482 is not issued to non-US site. Thank you.
Thank you for that clarification. Another question that came for you. How long does it typically take to receive an EIR after the inspection is finished?
Great question. So this depends on the resources available to the agency. We generally strive to make the inspection report available within 90 days at the closeout of the last day of the inspection. And we inform the site at the closeout of the inspection to say those are time frame and where they can contact the agency at if the EIR isn't received until that time. Please bear with us though that time to time we may not always make that 90-day because of a lot of priorities and resources, but we do strive to provide the report about 90 days after the closeout inspection.
Thank you for that response. Next question for you. Is it mandatory to provide written response to Form 483 on the observations which are already clarified or discussed orally during inspection?
Great question. So the response to 483 or written response to 483 is not mandatory. We are giving the inspected entity an opportunity to be able to provide additional information that could potentially addresses the concern that was issued in 483, and that's where we are giving 15 business days for that response to be submitted. However, it's not mandatory. Although it's not mandatory, it's strongly recommended because, again, that provides additional information that may not have been available during the inspection that may address some of the points that was made. In terms of if it's clarified during the inspection verbally, whether the information needs to be submitted, it's kind of going along the same line of thinking that it doesn't have to be written, and as I said, it's not even mandatory. However, during the inspection, the inspected site might have a limited window to be able to provide something in a more relatively short time frame. And if you can find some additional information that actually helps the situation to be resolved, it's all the more beneficial for the site to be able to do that. So just to make a recap, it's not mandatory, but even if it was discussed during the inspection, it is encouraged to provide those information in a written response within 15 days. And the center will consider those responses as a part of the whole evaluation of the inspection. Thank you.
Great. Thank you for that information. Next question for you as well, Dr. Cho. What is Form 483 and in which situations is it issued? And can you provide at least one example?
Great question. So Form 483 is a inspectional observation. The 483 just stands for the number is given to that form. As you might imagine, there are lots of forms in the government situation. So that's the name of the forms. But essentially, the intent of that form is to issue, to let the site know, inspector site know about the inspectional concerns. And just be aware that objectionable condition is based on the judgment of investigator, and that is not final decision of the agency. Once the observation has been issued and the written response from the inspected site is received, and we look at also the exhibits that was collected during the inspection, the whole thing is getting evaluated. And that leads to the final determination of that objectionable condition. So this observation is at the time of the inspection to the judgment of the investigator. And in terms of the example, I believe that if you go to the FDA website, there will be a template available, a template of the 483 available. So if that's what you are referring to, I think it can be viewed in terms of format and the content that goes in. The actual examples from the inspections, I believe those items are FOIA-able. So if you submit the FOIA request, I believe those can be accessed if you are interested. Thank you.
All right, thank you. We'll do one more question for you, Dr. Cho. Does the FDA issue establishment inspection reports or EIRs for inspections conducted remotely under RRA?
Great question. So as the EIR Establishment Inspection Report stands that it has inspection in the EIR, right? So, and we have discussed that RRA is not a form of inspection under our current paradigm. Therefore, we will not issue an EIR because it's not an inspection. Therefore, the establishment inspection report will not be there. However, we do provide a report of RRA in a very similar pattern and the content as EIR. We call it RRAR because it's RRA report. A lot of R's in the word, but we will provide a report, and the time frame for the RRA report is a similar, comparable to EIR. It's just that the name is different. So again, this is one of the things that we provide to the inspector site after a certain time frame after the closeout of the RRA. Thank you.
Great. Thank you again for all the information you've shared. We'll now move on to the questions that came in for Dr. Seaton. First question, Dr. Seaton, are there ever cases where FDA will investigate non-clinical or non-GLP research? For example, if it supports GLP data in a submission?
Thanks, Nora, and thank you for the question. Good question. The only time we would be looking at non-GLP studies on an inspection would be the animal rule inspections or the animal rule studies that I mentioned before. Otherwise, if it doesn't fall under the scope of GLP regulations, we as FDA inspectors don't have authority, so we won't be looking at that on inspection. Thanks.
Thank you. Could you also talk about regulations versus guidelines or guidances?
Sure, thanks. Another good question. Regulations, I mentioned 21 CFR Part 58, are things you must do in terms of GLPs. Guidelines and guidances that the FDA publishes are the agency's current thinking and best practices, but they are not enforceable on inspection. So you shouldn't expect to see something from a guideline on a 483, for example. So again, regulations you must do, guidelines and guidances are FDA's current thinking on a particular subject. Thanks.
Thanks for that explanation. Another question for you. Is there a database of GLP site inspections and results of those inspections?
Thanks again for the question. So there is a database, but it's such a long website that I'm just going to suggest that the questioner search for FDA non-clinical GLP inspections, and the FDA website has an archive of previous inspections where you can see sites inspected and the outcome in terms of NAI, VAI, or OAI. Thanks.
Thank you for that information as well. Another question that came in for you. Is a list of computerized systems and validation status requested during GLP inspections?
Thanks again. So partly yes, a list of the equipment used during GLP studies is requested. It's part of the current compliance program for GLP, so that will be a request that you receive. You might include on there the validation status. You will be asked, and we will probably look at the select one or two validation reports to audit. So be prepared for that. Thanks.
Thanks for that response. Another question for you, Dr. Seaton, regarding the master schedule. Does FDA typically want to see all studies included on it, or just the GLP studies to assess workload?
Yeah, thanks again. It's a good question and a question we've heard before. We want to see all studies on that master schedule. As you might imagine, assessing workload, some labs might do 50% of their studies GLP and 50% non-GLP. Well, if you have a study director that's doing half and half, then if we only see the GLP studies on a master schedule, we're only seeing half of that study director's workload. We want to make sure that they have the time and resources to address anything that might occur on a GLP study. They're the single point of contact. They're in charge. They have to know what's going on during that study. So when we're looking at that master schedule, we want to see everything that they're involved in. So short answer, GLP and non-GLP on the master schedule. Thanks.
Great, thank you. Another question that came in. Do you ever have inspections without a Form 483 needed, i.e. no deficiencies at all?
Thanks again, good question. And I'm going to assume you're not talking me personally, so I'm going to answer in general. Yes, there are times when a lab will not receive a 483. Those inspections will then be, most likely, will then be determined to be no action indicated. Thanks.
Thank you. We might have time for one more question for you, Dr. Seaton. If we haven't received an FDA GLP surveillance inspection for over 10 years, what should we do?
Thanks, Nora, and I think you should thank your lucky stars. So we generate our surveillance inspections based on studies submitted. So we might, there's a large number of GLP labs domestically and more in foreign countries. We can't get to all labs on a regular basis. I would say your name, your company is going to rise to the top. Keep doing the good work, adhere to GLPs, produce good quality data, and we'll knock on your door one of these days. Thanks.
All right, thank you so much for that response. And looks like that's all the questions we have time for. Thank you again to our presenters. We'll now hear from the discussion panel, which will be moderated by Dr. Tahseen Mirza. And now I'll hand it over to you, Dr. Mirza.
Yes, it's not noon, so still good morning. So, folks, the responsibility to run this panel session was given to me. My name is Tahseen Mirza. I'm the Associate Director for Regulatory Affairs from OSIS. We have 4 panel members in our panel today. These are senior members of our office. We have the director and deputy of each division, Dr. Charles Bonapage and Dr. Arindam Das Gupta are the director and deputy director of the Division of New Drug Integrity. We also have representatives from Generic Drug Integrity Group, both the director and deputy. We have Dr. Julia Cho, who's the director, and Dr. Kimberly Benson, who is the deputy director of the division. I have prepared a few questions to ask the panel members. Each panel member will get about 5 minutes to answer the question, and then time permitting, we may go back and get opinion of other panel members, and then maybe a second round of questions should there be time. The first question I prepared goes to Dr. Charles Boniface. The question is, what are the greatest challenges that OSIS will need to confront in the next 5 years? I know it's a loaded question. Take your time and try to give us as elaborate of an answer as possible. Thank you.
Sure. Thank you for the question. And as you said, it's a little bit loaded. The greatest challenges that OSIS is going to be facing in the next 5 years can probably be broken down into data integrity issues, auditing electronic systems, and development and, I guess, use of tools to detect misconduct, falsification of data. Regarding data integrity issues, we see data integrity with all types of studies, not just BA, BE, and GLP, but it goes across clinical and so forth. It's not uncommon that we hear about something every week, and actually we just heard about something very recently, which again impacts multiple studies across multiple regulatory agencies. So that's something which I think we're hearing about that more and more. As far as electronic systems, I think we live in a world where more and more of our data is captured and submitted, electronically. I think we are seeing less and less paper, and there's challenges with that. What we learned is there are numerous electronic systems. It's very difficult to be familiar with. Well, we certainly aren't familiar with all of them, but even many of them, it's very difficult to get access to them. Some of them are quite expensive. You need licenses. So it's very difficult to train staff. And so when we, do inspections, we are always encountering new systems. And yes, they're similar in some ways, but auditing them is challenging. Something that I think we are continuing to evolve is understanding and use of audit trails to make sure that any changes are captured, and we can go through that and verify, what was changed, when it was changed, who changed it, and why it was changed. And then the other thing is tools to detect misconduct or maybe falsification of data. It's interesting because the regulatory agencies are always trying to catch up. We're always behind the 8 ball. And so as we become, aware of a breach or misconduct, we learn about it and we try to apply it. The problem is, if you want to call it, the bad actors are they're ahead of us. And so it's very difficult to stay on top when we don't even quite know what we're going to be seeing in the future. And that's something that I think we're going to continue to develop. And I would guess AI is going to become, the use of AI is going to be increased, and it's going to become a bigger component in many of these data integrity detection tools in the future. That is all.
Thanks, Chuck. That was quite an elaborate answer. I know there's a lot you can talk about, but we have limited time. I will turn to other panel members to see if you have anything to add to this. No? All right. then I'll move on to the next question. This question is for Julia, Dr. Julia Cho. In the talk earlier today, it was said that OSIS inspection site selection includes information received from other foreign regulatory authorities. Can you talk a bit about OSIS collaboration with our regulatory counterparts, such as the scope and benefits of these collaborations?
Thank you for the question, Tassin. This is just a very pertinent topic in relation to the site selection, but overall our inspections strategies and things like that. So to step back, our office OSIS has been having this international program for some time by now, for quite a few years. We started as a small group, but then now that also has expanded to include agencies, quite a few number of them. And those examples that we are currently having collaborations include EMA, European Medical Agency, MHRA, which is the United States and the United Kingdom, the equivalent of FDA, WHO, as well as Health Canada. And those partners are some the agencies that we are currently actively get engaged with and working with. So if we are to ask like why we are collaborating with them and what's the benefit of it, like the first and the most, I think, things that we like to achieve by working with the industry, working with the international collaborators are the global collaboration and the harmonization of regulatory expectations. I know many of the sites who conduct the studies submit their data and an application in around the world in different regions of the world. And if different expectations from different agencies will make it hard to fit their application and study conduct in a different way. So by having this harmonization across different agencies helps the development of the drug and therefore helps the safe and effective drug available to patient. And that's the goal, one of the major goal that we are, why we are collaborating with internationally. And from the programmatic point of view, by collaborating with them, we shared information about inspection strategies and tools so that we can enhance each other's kind of the way that we audit for the data and study conduct for the data integrity. That helps really enhances the efficacy and the outcome of the inspection. And part of it is also sharing the intelligence of those sites that we are inspecting by sharing the inspection findings and what was kind of concern from other agencies' inspection. It will let us maximize whatever is available globally to detect where the problems are and try to ensure the data integrity. So those are some of the goals as well as the benefits that we are trying to achieve using the collaboration. So what do we do or how do we do that as a part of collaboration? We meet with the regulators routinely, usually based on a quarterly basis meeting. And then we also, during those meetings, we exchange inspection findings and time to time, if details needed, then we also exchange the inspection report. And we sometimes conduct joint inspection when there is a need for it. And all these activities are accomplished through the confidential agreement and confidentiality commitment so that we are like working under the available resources. We also do the joint conferences and then or joint publication or joint like guidances kind of things that we like to make sure the consistent message are getting out there, out to the world. And we have a time to time needed like strategy meeting when we try to establish certain areas of the topics and strategies for the inspections. Another area that we collaborate beyond those agencies that I mentioned is also we are actively working with ICH as a whole world kind of a harmonization strategy and harmonization activity. One of the recent example is the M10 guidance for the bioanalytical method validation, and that is also outcome of our active global collaboration. And there are additional like FDA level collaboration that our office participate to enhance our inspection program. So those are just few examples of our current activities, and that certainly feeds into our site selection and evaluation, as was talked earlier. So thank you for the question. Tassin, I think you may be on mute.
Yeah, I think I'm on mute. That was a lot of information, Julia. Thank you so much. If you may I ask a follow-up question? Is there any information that you do not share with our counterpart regulatory agencies?
Good question. So it's not that we do not share, but I think something may not be that relevant in sharing information is maybe specific for the certain application, because especially for generic drug, the comparator drugs are gonna be typically different, and whether it was certain ANDA or certain application may not be so relevant to them. The maybe focus is going to be how the study was conducted at a certain site that both of the agencies are interested in. So those are type of things that we may not necessarily need to focus on exchanging.
Thank you, Julia. I think the next question goes to Kimberly Benson, who's the Deputy Director. So the question is, are there any changes that the industry needs to be aware of based on the March 2024 guidance on handling and retention of BA/BE testing samples? So I'm sure a lot of people are interested in hearing from you. It's yours, Kim.
Thank you very much, Tassin, for the chance to talk about this updated guidance, the revision to the 2004 guidance. There's a lot to highlight, and I know I'm on limited time, so I will try and hit a couple of real salient points, but my advice is I highly recommend everybody go read this over for themselves. So I do want to note it's a draft. It's primarily a draft guidance. And that's important to note because when a final guidance comes out, there is a good chance that there's going to be some differences from the draft guidance just based on addressing public comments. So let's get to this guidance. One important change here is actually related to the reason that I just said it is primarily a draft guidance. I have to say it's not a phrase I've used often in my years at FDA, but specifically there's a section in here, it's section 4B, that's titled Quantity of Reserve Samples. And it, in fact, is issued as final guidance, which means it is for immediate implementation. What this section does is it describes conditions under which FDA generally will not take enforcement action against either a sponsor or a CRO if they retain less than the quantity of reserve samples from a BA/BE study that is actually specified in the regulation. So this section actually supersedes agency compliance policy regarding quantity of samples. And that was previously discussed in another guidance titled Compliance Policy for Quantity of Bioavailability of Bioequivalence Samples Retained under 21 CFR 320-38C. That was a guidance dated August 2020. And based on this section being finalized, the guidance, that guidance is hereby withdrawn. So what the guidance here in this section does, it goes on to note recommendations for in vivo studies having a minimum of 30 single dose or 3 multi-dose units, each of test and reference, in the original container, in total, across testing sites being retained, and at least 1 from each shipment of drug used in the study. As to in vitro studies, at least the same amount, so that's 30 single dose or 3 multi-dose from each batch used in all the in vitro studies conducted at the site, and again, at least 1 from each shipment. This differs from the regulation of 5 times the amount needed to conduct the release studies that are recommended. So this is essentially a less burdensome requirement, less numbers needing to be retained. This does apply to all reserve samples for BA/BE studies, including those that are currently, the studies are done and the reserve samples are currently held from previous studies. So There's much more in this section. So again, please do go in, read it, understand the current thinking of the agency as it applies to this topic. Another section I'd really like to highlight is actually just a single paragraph in the background section, but it gives some examples of findings that the FDA has seen around issues regarding regulations and retention of reserve samples. I always find it really helpful when, in addition to hearing, here's what our current thinking is, you also learn a little bit about, oh, and here are some problems we've seen in the past. So elsewhere in the document, you'll find more details and clarifications of these examples, but these examples that are listed there, and they are not exclusive to there being other examples, but one would be testing sites returning the reserve samples to the sponsor or manufacturer once they're done with the study rather than retaining them at their site. And I direct you to section 5 of this guidance, which would provide you more clarification on things like the roles and responsibilities of everybody involved in the study, whether it be conducted at a CRO, whether it involves SMOs, and whether the study is done by the sponsor or the drug manufacturer. I want to add one thing that's not stated in the guidance, but I have personally seen a number of times, and that is when a testing site sends the reserves that they have onto another site where additional studies are going to be conducted. An example of this would be a site is conducting in vitro testing and they have reserve samples. Rather than retaining them at their site, which is what they are supposed to do, they send them on to another site that is actually conducting either additional in vitro or more likely in vivo testing for the same drug and the same application. As with that first example, where the testing site returned it to the sponsor, in both these instances, it was the site's obligation to retain those reserve samples at their site. Another example is that we've seen is sponsors, manufacturers, SMOs, contract packaging facilities, where they actually are doing the designating of the test and reference units to be given to each subject. By doing this, they're actually precluding the testing site from randomly designating the reserve samples as the regulation requires. So I'd point you here to section 4A in the guidance, which gives you more information on sampling techniques, and again, notes the importance of the testing site being the entity that randomly selects the reserve samples. And that section also includes the nuance that occurs when the study is a blinded study. And the final example in the guidance is when you have a multi-site study, which is common in a comparative PD or a clinical endpoint BE study. So they're multi-site, they're blinded studies, and they tend to be conducted under contract with an investigator, a university, a doctor. So it's a doctor's office or a doctor's clinic. What FDA has seen in the past is that these clinics or offices actually don't realize or consider themselves to be the CRO, but they are. In these studies, they are the CRO, which means they are required to retain reserve samples for the study that they conducted at their site. So again, I'd point you to section 5 for more information on this. So there isn't time to walk through everything here, but I do want to say that one of the things that I really like about this is that while it's not providing a lot of brand new stuff, it's really clarifying things from the 2004 study. Besides adding in a number more examples, it goes into more detail and elucidates some of the things that maybe were unclear in the 2004 version. One other thing, I know I'm taking up a lot of time here, is that it stresses the importance of documentation, okay? So FDA recommends some best practices about documenting your detailed plan. How are you going to handle retention reserves? Have that in the study protocol. Describe the procedures. Describe what gets retained. Recommend that you include in the protocol and the study report the method for randomly selecting these samples, the testing site staff who are responsible for that, the total numbers that have been retained, the number of shipments. Important to know the number of shipments that the test and reference drugs came to the site in, the number of samples that are retained from each shipment, There's additional comments about the importance of documenting the storage of the retention samples, the reserve samples, the access to them, possibly transferring them to another site. How did you do that? Document, document, document. If you have these detailed, thorough documentation relative to all the steps involved with the reserve samples, it's my personal belief that a lot of the questions that the agency might likely have would already have been answered by your documentation. So overall, again, it's a great guidance in that it clarifies things a little more. There's no ability for me to touch on everything here. So highly recommend everybody go read it, and also remind you to also go read when the final version comes out, because as I said, the final version often changes subtly, at least from the draft. So thanks again for the opportunity to talk at least a little bit about this revised draft guidance.
Wow. That was quite a lot there, Kimberly. I hope everybody found the information useful. As we speak, we are also collecting comments to this draft guidance and providing it requesting it to be included in the final. I think industry has provided questions as well, comments as well. So when the final guidance comes out, it will have all the information that Kim was talking about. Thanks, Kim. So we have about 3 minutes left. The last set of questions go to Arindam Das Gupta, who's the Deputy Director in our office in the Division of New Drug Integrity. He has a two-part question. The first question is, what are the highlights of the new draft data integrity guidance? And the second part of the question is, what does data integrity refer to in the new data integrity draft guidance? So what are the highlights of the guidance, and then what does data integrity really mean? So there are two-part questions. Arindam, it's yours.
Thank you, Tashkeen. This is a great question. Again, like Dr. Benson, I would like to point out that this is a new draft guidance, and like all guidances, this is out for public comments. So this is a draft guidance. It's out for public comments. And when finalized, this will represent FDA's current thinking on this topic. So the new draft guidance on data integrity is for in vivo bioavailability and bioequivalence studies. And this draft guidance emphasizes the importance of robust systems and controls to ensure the accuracy and reliability of the data. This also includes stringent controls over electronic records and audit trails. This draft guidance also highlights the need for comprehensive staff training and clear documentation practices, sort of the parts which Dr. Benson also mentioned. Practices to prevent data integrity concerns and ensure compliance with regulatory standards. The draft guidance also recommends that the sites conducting studies implement a quality management system. And the draft also goes into parts where they recommend that the sponsors, applicants, monitored the studies conducted at CROs, the things which the studies when they are outsourced. So coming to the second part of the question, again, I'll repeat the question. What does data integrity refer to in the new data integrity draft guidance? So in the new draft guidance on in vivo bioavailability and bioequivalence study, the data integrity is referring to completeness, consistency, and accuracy of data throughout its life cycle. So again, simply, it ensures that all data, whether in electronic or paper format, is attributable, legible, contemporaneously recorded, original, and ALCOA. So simply put, it is ALCOA. So again, going back, it should be attributable, legible, contemporaneously recorded, original, and accurate. So that's the ALCOA for you. And that it is maintained in a manner that prevents unauthorized access, alteration, or loss. So Thank you. Thank you again for this question, Tashin.
Great. I think we did actually perfect with time. The time is almost over. So what just remains is thanks, everybody. Thanks, panel members, for providing all this new information and useful information. And from the audience, I hope you found the information to be useful, and hope you're taking some messages home through this panel discussion. So take it away, Nora, from here. So thanks, everybody. Hopefully, we'll see you again in the near future. Nora will take over and try to wrap up the session.